very good. Wonderful to see you. Always.
Always pleasure, sir.
Good. So we have had a wonderful week, and
Yeah.
this is going to be the last session.
Yes, sir.
Good. Okay, so I'm around. I will switch off my camera and microphone for a while, and I'll get back shortly. Okay?
Sure, sir. No
All
problem.
right. Thank you, Rizesh. Bye.
Good morning, Professor Bruggeman. Are you able to hear?
Yeah, good morning. Are you hearing me? Or
Yeah, we can hear you. How are you, Peter?
I'm fine. How are you?
I'm
Hi.
good too. So I think it's morning for you.
Yeah, well, I should say good evening. Sorry. Yeah.
But it's okay. It's quite a big difference in time zone. Thank you for agreeing to deliver this talk.
Yeah, it's my pleasure. And
We'll start. I think there are 10 minutes more. We schedule the talk from 7, so we'll start from 7.
Yeah, can I just briefly share the slides to make sure that it's working well?
Yeah. Please do share.
Yeah, just.
Namaskar, sir.
Okay, and this is the wrong view. This is the wrong view, no? So this is the right view.
Yeah, it's right. Yeah, we can view.
Okay.
It's fine. Yeah. If you want, you can leave it like that also. Share. No problem.
Oh, okay, that's fine. Okay, I just got my watch that I have a clock, so I know where I am in my talk. Just a second.
Sure. Hello, Parasar sir. Good evening.
Namaskar. Thank you very much.
No problems.
हमारे बच्चे भी ज्वाइन कर लिए हैं.
Yes, sir. Nice.
It's a good opportunity for me.
Yeah, very good.
So Rita, it's just a question from timing. So like 50 minutes, 1 hour?
50 minutes, yeah.
50 minutes.
55, 50,
Okay.
55. It's okay, 50.
Yeah.
And then we'll take some questions, 5-10 minutes like that.
Okay, great.
Hello, Sunil. How's everything at your place, Pete? Is it fine or is still COVID period is ongoing?
Well, I mean, I think we were better in summer than now. I think it's getting a little bit more. Well, I'm starting to worry again. Because semester is starting in 2 weeks or so, and I think the cases are rising again.
How's the situation with labs? Are they start reopening or still partially?
Labs have been open for quite some time in our university. So I think we only shut down for 2 months or so, really.
Mm-hmm.
And then we had a large time restricted access, so less people than usual in the lab, but still we had lab operation. But this summer, I mean, we went back almost back to normal. We still had to wear masks, but things were almost back to normal. And now classes also will be in person in fall, but cases are rising, so people are starting to be a little bit worried. So how is the situation? Because the news we hear from India is not so great sometimes recently.
Yeah, we had a really bad patch in at least 1 month ago. Like the second wave was very deadly for us. But our vaccine program is going in a fast speed. It's quite accelerated now. So that's why this third wave we are able to at least tackle. It slows down significantly. And in our institute, we start calling students back in batches, especially those who are like graduating or in final years require labs. So they are coming in batches and quarantine, but slowly only because it requires a lot of quarantine and vaccination before they can move to normal rosters. So we'll still only 10%, I can say, what in function, 10% of our student return. Hopefully, there is a prediction of third wave. I hope will not have much restrictions. Let's see. It's still.
Yeah, I think, you know, in my opinion, whatever will happen, I think we will have to be careful probably for another 2 years or so. You know, I did travel, although for the first time internationally this summer, and travel experience is quite different than it used to be, I must say. Yeah.
So we have our mentor of this Camost, Professor Deshmukh.
Good morning, Professor Bruggeman.
Yeah, good morning. Very nice to meet you.
Yes, indeed. And it's such a great pleasure and an honor to have you with us. And thank you very much for accepting our invitation to be a member of the Scientific Advisory Council for Camost, and also for the invitation for this evening's talk.
Yeah, thank you so much. I look forward to learn also more about your institute and maybe opportunities to collaborate. So thank you.
Absolutely. A major objective of our center is to participate in various collaborations within and outside India. And we're looking forward to that. And when I joined this meeting, I noticed you were talking about COVID, and that has really slowed us down and created so many difficulties. You mentioned the difficulty about travel, and we are all trying to adjust to that. It has been difficult, but I think things are picking up now. But as you rightly pointed out, we have to continue to be very careful.
Yeah, and I think particular for new initiatives, it is challenging, I found. I mean, if things are kind of running smoothly before, you know, with some adaptations, maybe it's possible to keep running.
Very true.
But for new initiatives, it's sometimes critical.
Yeah, but our center is very fortunate that we have several very young scientists in the group, and they're very capable and also ambitious. So that has helped us keep some activities going and undertake some new initiatives. But we do have a long way to go, and let's see how things develop from here.
No, I like the idea. So, you know, in the US, we often, So in the US, we often, I mean, we need a lot of AMO data, for plasma science. And
yes.
In US, it's often not that the link is not so strong anymore, and you know, maybe funding in that area for particular data that's needed in the plasma community is not always so significant. And I think it really slows down progress in our field, because for models, we need a lot of that data, you know, electron cross sections and all kind of things that sometimes, particular for new applications, it's not only science driven, but also application driven that are needed, that are just not available. Yeah. And it's sometimes a difficult message to get across to funding agencies, I find personally in the US. So this is really a great initiative in my opinion. And, you know, if you could, you know, having plasma, having AMO specialists in the same center, that would be really, I think, excellent and a lot of opportunities.
Yes. So we are very optimistic about how we hope to grow, but it's only time that would tell. So, but we are very encouraged by the beginning of our young group of scientists have made in the community.
Yeah, this is great. I look forward to hear more, in the coming years.
Have you visited India anytime, Peter?
Well, no, actually, no. I was planning to come for a plasma meeting earlier this year. So I was intending, it was in May, I think there was a plasma meeting, and local, national for India. But okay, unfortunately the situation didn't allow, so it was a virtual meeting. Yeah, I have never been to India. Yeah. So.
And then perhaps once the corona and everything is normal, then please plan sometime and put us also sometime in the agenda to visit.
Yeah, I would be really interesting to visit and also.
Yeah. Please do include Tirupati on your very first visit, and of course on the subsequent ones. But please make sure that it is it's a point to stop by on your very first visit.
I will do. I make that happen. Yeah.
Yes, that's great.
Deshmukh sir, we may start. It's we hit 7, so okay.
Yeah, maybe another couple of minutes, Ritesh.
Okay, sir. No problem.
It's
yeah, I think we can wait for a couple of minutes more.
Yeah, sure.
Yeah. So it's almost time, but maybe we can wait for a couple of minutes.
So Ritesh, you are now back in India like 2 years now, or how long is it, or is it longer already?
So I left Luke in 2016, University of Montreal, and then I was in Weizmann Institute for a couple of years, and after that I returned India. In 2018, I joined here.
Oh, okay. Time flies. Okay, sorry, I thought it was not that long. Yeah.
Yeah, it flies slowly.
So, Professor Bruggeman, if you and Ritesh are ready, I guess we could start the proceedings for the evening.
Yes, sir.
Okay, this is fine. Thank you.
Yes. So, good evening to everybody in the audience, and a very hearty good morning to Professor Peter Bruggeman, our speaker for this evening. It's morning in Minnesota, from where Professor Bruggeman has kindly joined us. I will first like to thank Professor Bruggeman for accepting our invitation to deliver a talk this evening. And I'm very pleased to welcome him also to the Center for Atomic, Molecular, Optical Sciences and Technologies, because he has also accepted our invitation to join the Scientific Advisory Council of the Camost, which he did rather recently. So thank you very much, Professor Bruggeman, for that. This has been a wonderful evening, wonderful week for us, because the center is completing 1 year since its inception in last August. And we decided to celebrate this as an anniversary colloquium series. So we began early this week on Monday, on 16th August, with a very nice talk by Professor Ravindra Kumar, and he talked about extreme states created by lasers, and he talked about plasmas, and he in fact commented that it's rather rather nice that the first talk and the last talk, which is this evening, which is Professor Bruggeman's, also overlaps quite a lot with plasma physics. And then our next talk was by Professor Vijay Raghavan, also from TFR, and he talked about building quantum computers. The third talk was on Wednesday by Professor Urvashi Sinha, and she talked about photonic quantum sciences and technologies. And yesterday we had Professor Dimitri Butkar, and he talked about physics and sensing applications of NV centers, nitrogen vacancy centers in diamond. And this evening we have Professor Peter Bruggeman from Minnesota, who is very well known, of course, but let me invite Ritesh to introduce him. Ritesh?
Yes, sir. Thank you, sir. So good evening to everyone and good morning to Professor Peter. It's my honor and privilege to introduce Professor Peter. Professor Peter J. Bruggeman is Professor and Associate Head of Mechanical Engineering at University of Minnesota. He is currently the Director of High Temperature and Plasma Laboratory and Associate Director of the Department of Energy Center on Plasma Interactions with Complex Interfaces consisting of 8 institutions. Professor Bruggeman's research mainly focused on the fundamental physical and chemical processes aspect of low temperature non-equilibrium plasmas, which is currently being employed in many novel applications consisting the area of environmental, biomedical, renewable energy, and agriculture, food processing, like that. His research findings, research accomplishment is enormous to mention here. But just to mention few, his research has been recognized by several awards, including most recently the 2018 Peter Mark Memorial Award of the American Vacuum Society and the 2020 George W. Taylor Award for Distinguished Research of the College of Science and Engineering, the University of Minnesota. He is currently the section editor of low temperature plasmas of the Journal of Physics D, Applied Physics, which is a very leading journal in plasma community. He also served on the committees charged by US National Academies with the decadal studies on plasma science, Plasma 2020, and co-edited the Plasma Roadmap, 2017 Plasma Road, giving direction for the future development of the field in low temperature plasma science and technology. Professor Bruggeman is also an elected member of Board of Directors of International Science of Plasma Chemistry. Professor Bruggeman has authored several research papers in leading journals of plasma physics. Apart from his research papers, I would really want to mention his review articles, which are very informative, and they all are in my list. And whenever anyone joins my group, I always ask them to read them. They are very informative review articles. Professor Bruggeman also currently involved in, he recently started one international low temperature plasma community along with Professor Mark Kushner. And the community's main objective is to, especially in low temperature plasmas, this forum is try to communicate, collaborate, and try to make a platform for the researchers who are working in this field and also the researchers who are interested in this field to make them familiar and give them platform to work on this. So with this short introduction, I'm really sorry not to mention a detailed thing. With this introduction, I would like to invite Peter to give his talk.
Thank you very much, Ritesh, for this too kind introduction. Well, thank you all for the invitation to be able to present or to promote our field, I would say, because it's a very general presentation. And also congratulations for the first anniversary of your center. So I've seen some of the talks, and I apologize not to be able to actually attend every morning due to a lot of other engagements. But I've seen that many of the talks kind of focused on one topic or one kind of application. So, but I really chose to, talk about our field that we kind of refer to as low temperature plasmas, although not everything is really a low temperature. So I will highlight this and really highlight, some of the capabilities we have. Within the field, kind of the questions we have, I think, for future research that are, I think, really interesting, potentially also for your center. And also the exceptionally broad range of applications, I think, that you can address with low temperature plasmas. And I would like to make a case of that in this presentation. Okay, let's see how. Okay, so at University of Minnesota, I just wanted to briefly mention, so we have the high temperature and plasma lab, like was mentioned in the introduction. And that lab has actually been around since '64, so it's much older than I am actually. It was established by Professor Fender, who was a National Academy of Engineering member. And over the years, I think there has always been 3 faculty in that laboratory. We have kind of an activity of about 25 researchers, over many years in the area of low temperature plasmas and plasma technology. So when I talk, and I know that some of the audience might be less familiar with the topic, I kind of want to frame it. And like mentioned, you had already a talk about plasma physics. That was what we would call quite extreme conditions, inertial confinement, fusion kind of conditions were the first presentation. And so the area of temperatures and densities we are calling or discussing today is actually a subsection of what you would kind of find in plasma section, and kind of indicated here in yellow. Now, although it's a subsection, we're still spanning 12 orders of magnitude in ionization degree. And as you know, in plasmas, the presence of charge is kind of a key determining property of plasmas. And we are spanning about 12 orders. And that's also the reason why the potential applications, okay, because of the hugely diverse properties, with these 12 orders of magnitude in ionization degree, is also equally huge. Okay, so it's not that we always using the same kind of plasma for every application. And then maybe very, recently, more and more recently, most applications of these plasmas, let's say 90%, are really related to producing of reactive species. And so the reason for that, why this is unique, is that we have a non-equilibrium. I will get back to that briefly in the next slide, where you can actually generate these reactive species or get reactive species distribution out of equilibrium that normally you would have very high gas temperatures to achieve that you here sometimes can even create it at room temperature. So that opens kind of a large range of new applications, particularly for treating heat-sensitive substrates or materials. And so why is this? Well, okay, low-temperature plasmas are by definition, well, almost in most cases generated by electric fields. And so your electric fields are accelerating some electrons that are already present in the gap, in the gas, to high energies. They are causing ionizing collisions. And so the energy you put into the plasma is this, through this electric field, and is mainly going through the electrons. So you're not directly heating with your energy source the gas molecules. And the case that if you apply this in the correct way, you can actually avoid to get equilibrium. And the reason for that is that the energy transfer of electron collision to neutral particles for gas heating, as many of you know in the AMO community, of course, is not so efficient, then you would have the energy transfer from an electric field to an electron. And so that is kind of the key, because this happens on different time scales, that you can create this non-equilibrium. It's kind of the key defining property for low temperature plasmas. Nonetheless, I want to highlight this, and I want to actually highlight this with a few examples that we studied in our lab over the last 60 years. It doesn't mean that it's low energy. Okay, this is really important to highlight. So, although thermal plasmas, so they can be 10,000 Kelvin or 20,000 Kelvin, we still call them low temperature plasma, the distinction with fusion that is maybe at much higher temperature fusion plasmas. And so that means that even in these kind of conditions, when they are electrical driven, you can actually generate very hot plasmas. Well, we, contradictory, actually still calling them low temperature. That was kind of a focus of our lab, up to maybe 20 years ago or 10, 15 years ago. It has many of applications in industry. Now, more recently, we are looking to low pressure plasmas. There, the collisional processes are slower, so the energy transfer to the gas is slower. You can easily make them non-equilibrium, has many material science applications. Semiconductor industry is built on that. And so, there are many examples in industry there. And then more recently, there has been maybe the last 20 years or so, a strong focus on really generating non-equilibrium plasmas even at atmospheric pressure, motivated by environmental applications, medical applications, where the requirement to work, for example, in atmospheric pressure air. And so that's a lot of engineering to design these sources, but it is possible to actually run them again at room temperature or close to room temperature, while still generating a lot of reactivity. Now, I wanted to highlight that because you could think about, okay, what does it really mean? You kind of maybe keep close to room temperature, maybe kind of 1000 Kelvin. It's not uncommon for some plasmas that we study. But what does it mean about power, and particular power density? Now, I like to show this in typical presentations are a bit broader, a comparison with the fusion plasma, tokamak, like all of you I'm sure are familiar with. And some of the plasmas we use, this is a filamentary plasma you see here. The length scales are highly different. So in the tokamak, this is several meters in cross section. The filamentary plasma show here, maybe it's the filaments might be 1 centimeter long, but the filaments itself are probably 10 to 100 micron in diameter. So the volume is much smaller. So of course the power we put in there is much smaller. If you look to power density, in many of the discharges we study, the power density can actually be higher than in a fusion plasma. So I think that highlights that the reactivity is really coming from this high power density. So, we can get still this non-equilibrium. Because of the small scale, you have a huge amount of energetic losses, heat, particle losses at the boundaries, and that kind of lead to potentially even having very low temperature plasmas at relatively high power densities. Yeah, so that's really where this reactivity is coming from. Now, for the applications, in a couple of, it's probably more than a decade ago, the US Academy of Engineering had 14 grand challenges for engineering, basically aligned with societal challenges we have. And if you think about this, what low temperature plasma can contribute, in principle, we can contribute to any of these challenges, for example, indirectly through advances in, for example, semiconductor processes that enables computers, your mobile phone. But even directly, even by half of these challenges, I think there is something that low temperature plasma can really contribute. I mean, I'm not going to go to each of these examples here, but I will highlight a few examples in my presentation about this. Now, I think it was mentioned also in the introduction. So in the US, we had the Cato study that, if we kind of look forward, might be also interesting. It's kind of a book written by a committee charged by the National Academy. And so one of the chapters deals with low temperature plasmas, looking back, really kind of illustrating how important it was. Like I mentioned already, the semiconductor industry, we wouldn't have computers without low temperature plasma based technologies. Really made huge contributions, but we also see many opportunities. And some of these were highlighted in the study. In the area of healthcare, agriculture, food cycle, really material science, the next generation. If you go for quantum computing, there is a lot of interesting science, but also making of these materials might actually involve plasma processes or low temperature plasma processes. Particularly in the US, and I think around the world, there is a huge interest in new chemical processes that may be not based on heat, but on electrical energy that could exploit renewable energy. We also see there a lot of opportunities for low temperature plasmas. And then, I think plasma has really enabled space exploration, satellite propulsion, and other things. Safeguarding communication infrastructure this way. And I think, there's a big, while I'm not working in this area, there's a big area of research also related to plasma thrusters, for example, that continues having great developments. So what I kind of going to highlight is really the reactivity of plasmas for different applications, talking a little bit about kinetics. And the reason why, the case I want to make that really scientific understanding of these plasmas is crucial is because of their complexity. So I just want to show here an example from a model where we look to This is just a few of the species we are producing in the right graph here, of reactive species that, for example, can be produced in an air-containing, humid air-containing plasma that is used for biomedical applications. And you see the huge amount of variety of different species. So if you really want to understand the kind of processes, you really need to understand the kinetics of these plasmas, even the electron kinetics, how this is translating to dissociation of molecule producing reactive species. And this is really crucial. It's not so one species you're forming that typically does the job. There are also, I mean, you have electric fields, there are charged particles, there are photons to consider. So there is many aspects to that makes the process extremely complex and on the other hand, of course, very interesting for the scientist. Now, you could say, well, okay, ultimately this is an engineering problem for the applications. While I agree, I think particular for plasmas, this might actually not be the way forward to just look at this as an engineering problem. And the reason why is that, the control lobes you have is basically the application of energy, the electric field you apply, maybe the gas composition or the pressure, are not really one-to-one linked to the outcome you want, which means certain species that are beneficial for certain applications. And the control lobes are really, it's a highly nonlinear system, and so kind of looking this as a black box, with some studies done initially when new applications are being developed, is often very time consuming. And so I think there is a huge need in our community for building predictive models. And if you want predictive models, you really need to kind of a full understanding of the underlying plasma processes. And I think this is where low temperature plasma science is really coming to play a huge role. And this is extremely important, I think, because again, I mentioned already this broad range of parameters, even within the subsection of plasmas that we call low temperature plasmas. It's just too much to really explore without having a scientific framework. Now, these are some examples and just kind of illustrating on the broad range of applications that people have been working on in the last 10 years. So you see here an example of water treatment. I think very novel there is that plasma seems to be the leading technology for addressing new emerging pollutants that are being recognized in drinking water, like PFOA, for example, where plasma seems to be outperforming any other technology that is applied to that. People have shown very impressive results on wound healing with plasmas, using the reactivity of plasmas to disinfect wounds, but also somehow stimulate wound healing. Material processing has been important for decades, but recently, going to smaller materials, 2D materials, atomic layer accuracy is sometimes needed. And I think plasmas have been shown to be able in well-controlled conditions to go to near atomic layer etching and enhancing the accuracies and reducing the sizes of semiconductor chips, for example. And then, very much currently in the attention is, of course, decontamination, where plasmas have been shown, even on virus quite recently, to be effective. What are we really looking at? And I think interesting thing is that we are looking for selectivity. Okay, so I just want to give an example of for example, biology. It's relatively easy to kill bacteria. You just take some bleach and scrub the surface and it's killed. So that's not the application that plasma is going to be used for. The application is, for example, doing this in a wound where you want to treat it at a certain threshold that it's effective enough to actually do something beneficial without having a negative impact on the wound tissue, where you cannot just pour some bleach because that would be a disadvantage or highly disadvantageous for the regrowth and the healing of the tissue. So this is the kind of thing we are looking at. So, and I think there are three areas that I kind of mentioned earlier from the decadal study where this is playing a role. And so kind of the or the conversion of natural feedstocks or waste materials into valuable materials. So very selective chemistry is a key science question. How can we do that with low temperature plasma or stimulate that with low temperature plasma, for example, combined with catalysts. You know, the 2D materials, like I mentioned here, is a nice example from colleagues in the UK. For example, in the oxidation of graphene, you know, can you do this accurately without damaging the materials? Can you actually control the amount of oxidation? So this is kind of the selectivity what we are looking for, and I think what is really needed to have a really detailed understanding. So this kind of comes me to the topics I really want to address today. So I want to talk a little bit about plasma kinetics that underlines all these reactive species, kind of the challenges we have, which is often, I think, related particularly at higher pressure, where I will be putting the emphasis on self-organization also in these plasmas, which is, I think, an intriguing physical phenomena. There is a lot of questions also related to transport. You know, transport of species, particularly for applications, because most of the applications in the plasma is in the bulk you produce the chemistry or the species you want. But ultimately, you need them at a surface to do some surface treatment, for example, and that transport is often quite complex, particularly coupled with plasmas. And then I would like to show some examples of plasma material interactions that we are starting to get some understanding. But you know, that are really, you know, quite complex, even including some biological matter. Okay, for the first one, so you know, self-organization, you know, in the broad meaning of the term, is quite common. And so you see here a regular glow discharge. So this is kind of the discharge like a TL tube for lighting in rooms, maybe in university halls still present. You know, you will have in such kind of lamps always some structurization due to the redistribution of charged species. So this is kind of quite common. However, you know, particularly at high pressure, you would not expect, because of the high collisions, that you will still get very distinguished patterns in plasmas. And you know, this is to some extent true that you wouldn't see this in the bulk of the plasma, where self-organization at low pressure would often happen. But really at interfaces like semiconductor interfaces, metal interfaces, liquid interfaces, and you see here several examples from a review paper, you can have many, many patterns, which is kind of surprising. And you know, studying these plasmas actually doesn't make it easier to have this kind of patterns, even for models. Now, what is really happening here, you know, it's actually, without going maybe into much detail, it's a Turing instability that is occurring. And it's basically a reaction diffusion equation related to basically current density is one of the drivers, and voltage drop that drives the ionization, for example, in that region. And so for some conditions, you can actually get instabilities just based on the basic equations of the system. Yeah, so this is kind of known. I mean, you know, in reality, I mean, we've seen many conditions that actually I don't think explained based on this basic phenomenon. On the other hand, you know, if you go to higher pressures again, and I think this is the area where maybe there is less fundamental understanding. I think low pressure has been studied in much more detail. You also get filaments. I mean, even if you look to our atmospheric phenomena, the aurora is relatively diffuse at low pressures, high in the atmosphere, while lightning at atmospheric pressure is certainly highly filamentary. So, you know, this is kind of a study is well known. You know, the effect is basically you form streamers, and that means that in this kind of situation, when the ionization is high enough, the space charge you build up is so high that the electric field introduced by the space charge of the plasma is actually dominant over the applied electric field due to the voltage, for example, across the electron. So basically, the plasma is producing its own electric field that is causing the ionization and its own propagation. Now, this is, of course, extremely challenging, but makes things more dominant, more complex, and really more complicated to study. And so this is an example from a couple of years ago that you know, we we try to get such a filament stabilized so that we have just one filament without branching, to look to really what kind of electron kinetics is going. Because if we understand electron kinetics there, we really understand all the subsequent processes, leading to the chemistry. And so this is an example. We spent actually several years on trying to really fully stabilize the discharge. You kind of see that this, is kind of a, this false color is light intensity. You see that it's not even attached to the high voltage needle. Basically, that's because the electric field in the head, because of the high electric field, you can see this as kind of a metal wire that is growing almost. No, if you want to think about the field configuration, that the highest excitation happens in the head. And so there were no measurements of this kind of phenomena because it was extremely difficult to stabilize. And you see here the first measurements with Thomson scattering of such a phenomena where, we call this a streamer, where the streamer head, we actually see the electron density distribution, and then also the electric fields, as a function of time when the streamer is basically passing the laser beam, where you see that electric energies of 4 eV are achieved. And where you see that electron density is going down very quickly. So what is kind of the key point here? This is a room temperature plasma that has electron energies of 4 eV. 4 eV is plenty to generate electrical fragmentation of molecules, and then lead to this kind of very rich chemistry that is useful for the applications. Yeah. And so, our understanding, and then of course complemented with modeling, really kind of helps now to, okay, how are these processes driven, and how can we kind of improve these processes based on a fundamental understanding? Now, if you look to surfaces, you can have the same phenomena on surfaces, and this is actually, very much less understood. And particularly here you can have diffuse, so kind of discharges, or something that even get, stars kind of shape, where these filaments are really forming patterns. And this is something we are currently also studying. Now, what I talk now, this last few examples were actually relatively low power density, temperature was room temperature. But you also have filamentary discharges that are actually very intense, maybe resembling more lightning conditions. And one of these examples is when you have a much higher pressure. So thinking about combustion conditions, for example, where people have studied that in a few atmosphere, above atmospheric pressure, or even in liquid. So the example I show here on the left bottom is a discharge in liquid. Now, in these kind of conditions, we have seen that ionization or electron density can be higher than typically the density at atmospheric pressure. So you can have a huge density. This is typically done with nanosecond pulse discharge. Power density is much higher than you get in a fusion reactor. And kind of interesting, if you look to the coupling parameter, so for example, warm dense matter, where you have a high coupling parameter, the interesting thing here, the temperature is quite low, okay? And so you can actually have a coupling parameter over about 1 in these plasmas, which is quite unusual for having low temperature plasmas that can reach these conditions. And this is kind of really, I think, quite unexplored. We're starting to understand, how this can happen, because these plasmas are really generated in a few nanoseconds. Okay, so you need to go from almost no ionization to almost full ionization in a few nanoseconds. And this is actually was remarkable for a long time. We didn't understand, and we kind of have some emerging understanding. Now, to conclude this aspect, I think that we kind of understand quite some of the basic mechanisms. We still have a lot of questions related to detailed kinetics sometimes. In some cases, we have predictive models. So this is an example here. And for example, helium, water, and really very diffuse academic, let's say, plasma parallel plate, you can model 1D. We have some good predictive capabilities. There are many conditions, very much interested for a broad range of applications where that's not the case. And there are different reasons. Often, for example, cross-section data not available, but also, not having some diagnostics for probing these aspects of these discharges that are important. And so typically 2 examples here from a diagnostic point of view is Thomson scattering. The electron energy, like I said, is often, around 4 eV for these plasmas. But ultimately, if you want to really understand ionization, most ionization energies are like 12 eV, and there's basically no diagnostics that can probe how these electron energies are actually behaving at, even at low pressures where you have probes. It's often challenging to reach this kind of conditions. And then another thing is like, particularly because of these patterns, spatial resolved measurements are often extremely important. And particular at high pressures where you have gradients on tens of micron or even smaller. This is quite challenging, for example, for laser diagnostics. And there's a lot of things still to be done there. Okay, so moving forward to plasma transport. And so I'm not talking here about transport, for example, in magnetized plasmas at low pressure, but I'm really talking here about transport maybe from the bulk of the plasma to the boundary of the plasma where processes or applications of interest are happening. Now, this is quite challenging. So this is an overview of some plasmas that are used at high pressure for many applications. And you kind of see, just looking to these pictures without going in detail, how different the different kind of plasmas are. So, the things we're talking about might be quite different from one source or for one type of plasma to another. But, just to talk about specifically one that has been extensively used in the last few years for biomedical applications. And you see here on the left is a so-called plasma jet. And so basically the challenge there is that typically when you use, for example, for biomedical applications, the plasma is generated away from the source, and then you have a jet, a gas flow that's actually blowing towards the source. So you have convective transport of some of these species, and then depending on their lifetime, they are actually recombining and might not actually reach even the substrate. But on the other hand, the plasma can actually also partly be transport, or the ionization wave can actually go down to the substrate as well. And then you can get a combination where you have a coupling between the plasma dynamic and the transport, which becomes quite difficult. I mean, just letting the last part away and just thinking about, okay, how can we do this? So we looked at that. I mean, again, a very academic example, the most simple example maybe just to get an idea of what the importance is of some of these studies. We're looking into a plasma jet. Now it's basically a tube. We generate a plasma in the center, and then this is blowing towards the substrate. To get some idea on how species are transported, we send in a laser beam through these plasmas, and in this case, we're looking to atomic hydrogen using 2-photon absorption, laser-induced fluorescence, and how these H radicals are produced, how they are transported, and how they could actually have an effect on the substrate. And if you look to this is a radio frequency plasma that is modulated. So this way we have like one-dimensional information on how these H densities are transported, are produced, where you see kind of the maximum, and then after the pulse, how they are being transported. And while they are transported, they are basically reacting away before they are reaching the surface. And so you can actually nicely visualize that. This is relatively simple. Of course, if you have more than hydrogen in there or complexer molecules for applications, things can become very difficult. And this example is probably the only example I'm aware of that you can actually solve with pen of paper and get the full dynamics with an understanding even of the electron temperature and electron density through the kinetics, following the kinetics of these pieces. Now, that is kind of probably the most simple case, but things become quite complicated when, like I mentioned, you get a coupling of the plasma dynamics with this neutral transport. So what I'm then talking about really is, for example, non-local electron kinetics. So what does this mean? Like in many cases, if the electric field is not too excessive, or the gradient in the electric field is not too excessive, the electron energy will, due to the collisions that happens in these discharges, particularly at atmospheric pressure, will be in good equilibrium with the local electric field. However, if you start to have huge gradients in the electric field or charge species density, that kind of assumption becomes invalid. And then you really have kind of runaway electrons, for example, moving much faster than the propagation of the ionization wave, kind of pre-ionizing the discharge and making it easier, for example, of the discharge to be formed due to these small fraction, but still carrying a lot of energy of highly energetic electrons. Another area where it's quite challenging is ultra-fast gas heating. So what is happening there is that I mentioned before, we can get very high powers, although for a short time, power density is very high. That leads to heating. So that is not heating due to electron collisions with neutrals. So the heating in that case is happening due to the electrons, for example, dissociating nitrogen, forming electronically excited state nitrogen. And they have a very short lifetime, particularly when there is a lot of, for example, atomic oxygen or O2, where they are being quenched. And the quenching of these highly electronically excited states leads to a huge amount of release per collision. And that is, the energy can go very quickly into gas heating, where you can have gas heating of about several hundreds of Kelvin to maybe thousands of Kelvin in a few nanoseconds. And so this is quite atypical for most systems. And then, of course, there is an expansion of the fluid. There could be shock wave formations and all kind of dynamics that are related to this very rapid changes in gas temperature with the plasma. So these are kind of really still a lot of challenges on trying to understand this kind of phenomena in low temperature plasmas. But as I kind of mentioned before, many challenges, and particularly if you're working on applications, are really related to interactions on surfaces, particularly how reactivity is being transferred. And so I want to highlight that with a few examples to kind of conclude my presentation. And so, some applications for materials that I want to highlight that people have been looking at now. Certainly nanomaterial synthesis is important. Biomaterials, bioactive materials, for example, for drug release or delivery has been really a point of attention to me. Materials which I already mentioned, atomic layer etching. So there are many kind of works going on in that area. I wanted to highlight an area where we kind of recently started to work on that I think is quite interesting also from a point of view understanding plasma kinetics, is the coupling between plasmas and surfaces, but catalytic surfaces. And that really kind of is driven by, can we do selective chemistry? If you heat a surface, and even with catalysts, you can get maybe more selectivity because you can do things at lower temperature. But ultimately, temperature or gas temperature is not helping to do selective reactions. And so we are kind of having the view that if you use plasmas, where you can kind of drive things at very low gas temperatures, and maybe combine it with catalysts that have maybe similar properties, but then on surfaces where it's maybe not always driven with electrons, but with surface reactions where things can also happen at a lower temperature. You might be able to enhance reaction rates. You might be operating at lower temperature in these processes. Can be higher selectivity, and maybe can actually really help to electrify the chemical industry. So this is kind of a very it has been around for some time, but in this context, kind of a new trust, I think, in our field. And so there are examples where, for example, for CO conversion, where kind of the plasma-based activation of a traditional thermal process actually showed really significant improvements. Now, this is actually quite difficult to study, and you can imagine, just to give you an idea, so these catalytic beds, all kind of particles, people are producing plasmas inside these beds. It's as inhomogeneous as you can imagine, so it's really difficult to study that. You have all kind of gradients, so it's really not possible. And what really kind of the trust we've been looking at is, can we produce the plasma remotely, let it interact with the surface, where we can actually really control the fluxes and also measure exactly the species fluxes going to the surface, what kind of surface processes are happening and what kind of resulting, for example, molecules that are being produced, and are they different than plasma? Are they producing at a different rate? So this is kind of the key big challenge in that area. Can we really understand that to fully exploit its potential? And what we've been building for that, is, for example, molecular beam mass spectrometry. We spend a lot of time, really trying to track all the dominant species that could be responsible for reactions on surfaces, and how these are driving, has kind of led us to development of new measurements of electronically excited states, developing techniques that allow to calibrate this, even ions, which is not so trivial in a mass spectrometer to calibrate, so that we can get a full picture of what kind of species are really going on the surface, and then kind of link this to surface diagnostics in situ, which is kind of ongoing. Now, why is this so important? Well, I mean, if you think about all kind of materials that are being used in plasmas, the chemistries that are involved or being produced in plasma, so the chemistries that are involved in this, many of these species, we don't even have data for electron cross sections, for example, of dissociation of ionization with these molecules. So there is a lot of data that's needed to really make a model. So really the only way to study this right now in many cases is, in my opinion, experimental. And so, if we could get, collaborations with the AMO community, on looking to, particular interests in certain species to try to advance, models in that area that are immediately applicable to important applications for material processing, for example, I think that would be a huge step forward. I mean, even thinking about diagnostics, so with mass spec, it's less of an issue, but if you look to laser diagnostics, many of these molecules and transitions, I don't think there is a lot of data available sometimes to actually probe them. For example, even with techniques as simple as laser-induced fluorescence. And I think then from a materials point of view, really integrating plasma models with, almost quantum models of materials and how processes are actually happening, I think is another frontier that has been really almost untouched. I think plasma models now really have coefficients, sticking coefficients in their models that are often estimated. Sticking coefficients are sometimes measured, but the accuracy of these coefficients are relatively low. In particular, when the interest becomes in predictive modeling, even of predictive modeling of surface processes, I mean, a better accuracy there, I think, would really go further, but again, would require a lot of insights in what really happens on these interfaces, combined with experiments as well. That is a solid material. Okay, so one step further, we did a lot of work on plasmas interacting with liquids as well, which is of course one step more complex. Although applications are really huge. I mean, I already addressed some of these, so I'm not going to repeat that. But something I didn't address, like liquid walls for fusion reactors, have also similar challenges there. There are, of course, liquid metals rather than, for example, water. Analytical chemistry is a community that has used a lot of plasmas for developing detection techniques and often also solution-based processes. So there is a lot of interesting work there, also often not so well understood, to be honest, but also chemical synthesis. There are many kind of applications that could exploit plasma liquid interactions. Now, what is so much more challenging there? Of course, you have the same kind of species fluxes, but the amount of, for example, having water, the key difference is there, of course, okay, water can evaporate, so you can actually have a huge amount of the liquid phase species that actually get into the plasma and really change your plasma. So you have this very strong coupling between plasma and liquid. but water behaves quite different, from a fundamental point of view of charged species going to the liquid, how charges are stored. I mean, solvated electrons would be penetrating on dielectrics, electrons would be, the electric solids would be only on the surface. So there are a lot of differences there on how the interactions between a liquid, for example, and a plasma would do. And then ultimately, if the process is really happening in the liquid, you still fully need to understand how these species are being transported. And because many of these species are short-lived, you basically have a boundary layer of the liquid that is extremely important in that process. That could be tens of nanometer to micrometer size. That is almost impossible to probe experimentally. Yeah. And the conditions there are highly unknown and might actually impact a lot of the things we are trying to do. Now we started to, again, our lab is always into trying to do very simple things to try to enable us to draw stronger conclusions than looking to very complex systems. And so we wanted to actually try to link, and I think that was probably for the first time, plasma kinetics and chemistry in the gas phase to whatever we're doing in the liquid. And so we are sending here, and I kind of like this experiment with small droplets going through a homogeneous plasma. We kind of apply our laser diagnostics on the plasma. We know OH radicals, for example, here is the dominant species. And then we can actually, load the droplet with some species. We can collect them. We can see what kind of reactions or quantify the reactions in the liquid phase. And we can actually try to make a model to bridge these two. So we've been doing that. And then interesting, maybe these droplets are water. They go through a plasma. The residence time is like maybe, 5 to 10 milliseconds. And they are not evaporating much. Okay, so we're working on very low temperature conditions where we can actually send droplets through the plasma. They are charging. We give them a sufficient kind of momentum so they can actually transfer through the plasma. And so, I mean, without going into much detail, we've been measuring the conversion. We've been modeling that process. And you can see here our comparison of the modeling of the decomposition, which we use here. Form rate is probably the most simple possible hydrocarbon that you can have in a liquid. The species coming from the plasma. And you can actually see, relatively good agreement. I mean, for the first time ever, where, without any fitting parameters, where a model is compared with an experiment in very controlled conditions, that we can actually, based on some simple chemistry, typically here OH-driven chemistry, predict also what or confirm, get excellent correspondence with the experiment on what kind of reactions we are introducing in the liquid. And interestingly, this is also quantitatively, and there is a reason why. Okay, so I don't believe we have excellent understanding of all these processes. There is actually a reason why we have excellent understanding. And in this particular case, the excellent correspondence between the model and the experiment is because this is transport limited, meaning that plasma produces abundant reactive species going to the liquid. They penetrate here, and you see, for example, the OH radical, on the order of, this is a droplet of 30 microns, so the order of 1 to micrometer. And basically, this is where the reactions occur. So your molecules that are in the liquid actually need to move to the surface to be converted, and then kind of move back once they are converted. So there is a transport limitation where actually the diffusion, because that's the dominant transport on these time scales here, is limiting the reaction. So basically, as long as we have enough radical species from the plasma, and as long as we model this diffusion properly, our result will always be correct. And it's most likely why we have so excellent agreement, because I'm sure there are some processes at the liquid that are occurring that we are not capturing in the model. But you kind of see here that it's also kind of an understanding of the dominant timescales and dominant processes that occur on these timescales that really can help a lot. Now, in state-of-the-art modeling, things are really complicated, and I think we are not there yet to kind of reliably do this, with the exception maybe of 3D models where plasma arc welding. And so this is also a plasma liquid interaction. This is actually has been reasonably done. The reason there is that the plasma model is, of course, much easier because you can assume almost equilibrium, and in these models this is done. And then you can, kind of model what the plasma is doing, how the molten metal and the evaporation of the metal in the plasma is impacting the welding process. And there actually predictability has occurred and has been able to, for example, improve welding processes. But in the conditions where I'm talking, the models are actually less developed because we have these non-equilibrium effects that are just way more difficult to incorporate in the models. And I just wanted to highlight, we have a lot of things going on in this area. Maybe also, kind of I am maybe putting too much time on this here, but the key question that we really want to address, and this is with a multidisciplinary team we have, it is called MURI projects, multi-university research initiatives. We're a group of colleagues from chemistry and from plasma science and material science, where we are trying to understand, and we understand very well, the non-equilibrium nature in the gas phase, how we can translate this non-equilibrium nature to reactions inside the liquid phase. And there is a lot of interesting questions related to electron solvation, ultra-fast reactions of pre-solvated species, how these conditions in the interface are actually changing that, which even involves a lot of calculations of new reaction rates, for example, and development of trying to develop new techniques to actually probe that interface. Okay, and then I think the ultimate complexity, and this is kind of the last topic I want to touch upon, is really when your plasma is interacting with biological living matter, and how does that interaction happen, and how does this impact actually a living organism that is changing because it is living in time. It's not a kind of innate surface. That's probably easier to understand from a perspective of material science or chemistry. Now, we've been, maybe lucky, I don't know. So we started working on plasma virus interactions maybe in 2013 when I joined University of Minnesota. And well, of course, now there's kind of a boom and everybody is interested in that. But originally I was interested in food decontamination. Again, the selectivity where you want to inactivate a virus but not impact the food. And that's typically for foods, of course, that are not cooked, that we were using. And so the biggest concern is norovirus in that area. And so we kind of showed that plasma is actually inactivating that norovirus quite effectively. But we were also more interesting fundamentally. Okay, so people are studying how plasmas interact with surface and how these surfaces can be etched. And so, but what is actually happening when a plasma is in contact with the virus? Are we just destroying the entire virus particle? And so here there are 10 images of such a virus before treatment and after treatment. And what we found is that actually quite remarkable. So when you treat a virus with a plasma, you don't kind of blast it till everything is gone. Okay, so very soft, and that you just have the minimum time you need to make sure the virus is no longer infectious. So basically what you do is you take the virus, you add it to cells, and you see that they are no longer attacking the cells in the cell culture, so they are no longer infectious. So what is then happening? And so remarkably, you see the blue arrows here. We see that some of the virus particles seem to be no longer infectious, but they seem to be intact when you look under a 10 microscope. And so that kind of triggered a lot of questions. So what is the plasma really doing here? And then ultimately what we looked at, and in the case of an oxygen plasma, to make things more simple, this is actually oxidation of the capsid that consists of proteins. And some of these proteins have the functionality probably to attach, have the functionality to attach to the cell. And maybe if you oxidize these proteins, they cannot fulfill this functionality anymore. And maybe that's the reason why they are no longer infectious. Because clearly we don't, some of the integrity of the cells is lost, but for some of them it's not. And then, that is kind of interesting, because if the inactivation mechanism is really oxidation, can we actually, due to a detailed analysis of the changes in these proteins, can we actually look to what species are responsible for the inactivation of the virus? And now in this particular case, in In an oxygen plasma, we've done a lot of measurements, but we've seen that a lot of these oxidation reactions can be ascribed to or ozone or singlet oxygen. And we actually shown that if you just blow ozone, the effectivity is really not as good as having also singlet oxygen, and kind of suggesting that singlet oxygen really here is one of the key species. So this is the first electronically excited state of oxygen, is one of the key species that the plasma produces for inactivating this virus. So that triggered, of course, a lot of studies. So can we really understand from a fundamental point of view, how we need to control the plasma to actually get efficient virus inactivation? And how does that link? So do we understand gas phase kinetics? So we measured singlet oxygen in the gas phase, we modeled that, and we kind of see very good correspondence, maybe not too much at the end, between the gas phase measurements and the liquid. So based on that, we understand transport. And then because these viruses were actually in the liquid, we also tried to measure the singlet oxygen in the liquid. We could actually find it back at significant amounts in the liquid, and find actually a correlation between the amount we found in the liquid and the inactivation amount of the virus. And then, with these proteomics results, where we actually track down the reactions of that singlet oxygen back to the surface of the virus capsid, that led to the virus inactivation. And I think, these are really kind of studies that I believe, really try to link directly, the plasma processes to the application in great detail. And I think can help us kind of move forward many of the applications in that area. Now, I talked about transport limitations, and I just wanted to show this because people kind of typically are very interested in that in view of the pandemic. We talked about transport limitations. And so typically it took us about a minute to inactivate virus on the surface, which is a bit long. And ultimately, particular for in the pandemic situation, aerosolized viruses are potentially a bigger problem, certainly for certain viruses. Now, that work was actually, again, done before the pandemic, but motivated by animal virus, transfer of animal virus. But if you want to do this in air handling systems, you need to kind of be able to inactivate, if you want to do this within like 10 milliseconds, okay? So this is about 1,000 times faster than we done on surface. And so motivated by these studies on these droplets, where we could actually, decompose a lot of the droplets in a few milliseconds, we build a reactor, put it in a wind tunnel, and we're able to show that actually plasma is able, if you send the gas stream through the plasma for a contact time here of 15 milliseconds, 15 milliseconds, fully inactivate the virus. And what you see here, this is the blue curve before and after. This is a log scale. So we have like 3 log reduction. And actually the remaining half log is the detection limit. So we cannot find any viable virus. However, just as I said, the plasma in this case is not a filter, okay? And this is kind of interesting, and we understand now why, because we are oxidizing the capsid, certainly in these, it's soft treatment, well, soft treatment, but I mean, it's not over treatment where you destroy everything. You see that actually when you look to the RNA in this case, a lot of the virus particles are still there, but they are no longer infectious. So it's a quite different mechanism than you would expect for filters. Now, we are trying to drive this. So this is some other project, multidisciplinary, and I think this is really one of the advantages of low temperature plasma science, and really one of the reasons why I'm so excited, because I can work with people from all different fields. And this is in collaboration with colleagues in microbiology, where we are trying to understand fundamentally how plasmas are not interacting with one virus particle, for example, but with a community of bacteria, which is called a biofilm, because that's really, these communities, they are having processes, chemical processes that they are communicating with each other, signaling processes. And so we are interested in, if this is really kind of a living community, how does plasma interact with a living community, bringing kind of the next level of complexity into that thing. Now, why is this important? Of course, this is funded because there is relevance for society. We shown before that plasma is able to really effectively inactivate MRSA, so antimicrobial resistant bacteria. And so there has potential if we can really understand that and kind of optimize that process. And so this is interesting. So as a plasma scientist, so the first results we got, and maybe kind of an example, I mean, maybe not as in-depth as the virus work that took us 7 years or so to complete, and this only recently started. But plasmas can do many things. And so here we just look to conditions where biofilm is kind of a very thin layer, hundreds of micron maybe, at the surface. And so when you have a liquid layer, what basically the plasma is doing is it's bringing species in the liquid, and you see kind of inactivation, which is the red curve. So the green means biomass, and if you see red, it means there is inactivation. So you see in the controls, the two left cases, nothing. You see that the plasma is inactivated. You see a lot of red. But remarkably, when you do it in dry cases, you see that there is kind of a depleted zone in the green, and there is no red. Well, there is a lot of red on the edges, where you're actually suggesting you are removing the biofilms. And so these are actually two different modes. Quite interesting, the dry case, what actually the plasma is doing, we have an oxygen plasma. The O atoms are etching away the biofilm, like they would etch a polymer, for example. And in the wet case, there is no etching because the species go to the water, you produce chemistry, and there is a chemical action of the inactivation of the biofilm. So even, very similar systems can kind of lead to completely different kind of processes that are enabling these biological interactions. But again, this is just initial work, and there's probably still several years that we will be working on to get a deeper understanding. Okay, but this, I would like to present you my conclusions. So, I mean, they are very general, but I think low temperature plasmas really had a major track record, I think, in the last decades for advances, and particularly recently, there are new areas that are being embarked, even making the field even much more multidisciplinary than before. For example, in some examples of health or materials that have been addressing. Now, from a scientific point of view, there is a lot of interest in this non-equilibrium, I think, and then still several questions, that we can kind of tackle. There are complicated questions, but it kind of drives, also the scientists in our community to look into these systems. Now, for applications, and many people do applications, you often have very complicated systems, and if you really get an understanding, I think it's really needed to have a dedicated effort to this, with kind of model systems that are highly controllable, that you actually can do detailed, I didn't highlight many of these things, but we do a lot of detailed laser diagnostics, that is actually enabling that. And that's also amenable for relatively simple models, that it doesn't make it too complicated to gain understanding. And sorry, and typically, I think for applications, I mean, I hope I convinced you in the beginning of the presentation, there are too many parameters that can change in the plasma to really, have scans of these parameters to optimize a system. So, I think people working on applications and industry really would appreciate more predictive capabilities than we have now. This will really require larger efforts than single groups, comprehensive coordinated approaches between experimentalists and modeling that kind of would work, for example, in centers like you have. And I think, really what has been, one of my great interests is bringing plasma science and plasma physics, across the borders and bringing together expertise from many different areas to tackle problems in our field, and also collaborating with many colleagues. And so we have collaborators in biology and in medicine, in chemistry, in material science. So, this is, it's interesting, I think, as a researcher to work in that area, because you learn new things from different fields, but you can also, I think, really much more strongly advance the field by these interactions that bring in new knowledge and new capabilities. And so with this, I would like to thank you again for the opportunity to, hopefully give you an overview and a flavor of low temperature plasma, and I would be happy to answer any questions you would have.
Hello, Sunny. Thank you very much for the wonderful talk. So it has been so inspiring, and actually it was like more of a question than those who have been already answered. Like there are so many open problems that have been discussed throughout this presentation. I think this will be very helpful for the students, those who are working on this field, and also those people who are doing similar research in AMO community. So I think now we can have a question and answer session. So I request the students to send their questions in the chat box, or you can also click on the reactions to raise your hand and ask questions for yourself. Okay, so now we have a question from Punit. So is there a way to apply post-plasma activated water for bacterial and biofilm applications?
Yeah, thank you for the question. Yeah, this is true. So, I mean, you mentioned plasma activated water in your question, and so that already suggests, of course, that once you apply a plasma to generate species in the water, that there remains some activity even after the plasma. Now, the activity remains. It depends on the kind of chemistry you produce, but particularly when you have reactive nitrogen species, there have been cases shown where the bactericidal efficacy is still present 1 week after it has been treated by the plasma. Now, one needs to be careful with that, because the chemistry will be different, and many of the species that are responsible for this, let's say, inactivation of virus or bacteria after the plasma has been applied, they continuously undergo reactions. So depending on the composition of your liquid, or even the temperature of your liquid, the lifetime of the species would be quite different. So I don't know if you can say it, it's probably not a way to sell plasma treated or activated water in bottles. Okay, the lifetime is going to be too short, but yes, there is a time where it is effective after treatment.
I hope that answers the question. So now there is another question. Thank you for the awesome talk, sir. I have a question regarding the plasma jet torch. I have seen this plasma jet torch in UFC, IPT, at IPR, but why they are not applicable commercially? Like we don't have such in many hospitals.
Yeah. So, well, this is a great question. So, there is one thing of showing great capabilities in the lab, and there has been clinical studies, and actually there are commercially available plasma jets that have been approved to be used, in a hospital setting, but there are very few. Okay, and so, for example, the kin pen from IMP in Greifswald, I think, has received approval to be used, at least in clinical studies. I don't know how far they are, but this is indeed not generally applicable in hospitals. This is still a research stage, and I think the key point here is really that it works. I mean, many people have shown that in several cases, there are very good results. The challenge often is that you need to show it is safe. And I think that is the key challenge for plasma, kind of also related to the understanding, we don't just produce one species. It's not just like radiation therapy where there's just one kind of radiation going to the wound. You have kind of a huge amount of different species. Maybe you produce ozone and other things that for a work environment can be problematic, that you need to kind of mitigate or have a solution for, to capture it, for example. So there are quite a lot of challenges to go to regulative procedures for approval of these things for a broad use in the hospital. Now, this being said, this is, I think, why there are not many, but there are a few cases that, this is being done now, or that I think, ultimately there are plasmas in the hospital. I just want to underline that for example, argon coagulator is actually pretty hot plasma that is extensively used in the hospital. Okay, maybe much hotter than the plasmas we talk about. But there are examples of plasmas being used, for example, for soft tissue in knee surgery in the United States. Is a company that has this and has been approved and is being used. So there are examples. But for many of the applications I talked here, there's really still research states and in the process of being worked towards approval to get it in the hospital. Yeah, so it's early stage.
I think in the healthcare sector, it will require a large number of tests for which patients have to volunteer and so on. So I think getting approval is going to be a big challenge in this.
It is very challenging, and I think there are. I know, for example, in Russia there has been a long tradition on anode treatment for wound healing. This is actually they use plasmas for that, but the plasma is not in contact directly with the wound, so it's a remote generator. That basically produces a low NO, and the NO is kind of brought to the wound. So there are examples, but yeah, I agree. The regulations are difficult. On the other hand, I mean, we've been focused on wound healing, to some extent, and the kind of problems that are there, particularly with MRSA, are huge in hospitals right now, and so the potential is there, I think, but it will take a little bit of time to get general acceptance of the technology, and probably a lot of work still from the community to show, like, this is working well, this is reliably working, and this is working safely. I think we have several examples, but maybe not enough to convince.
The safety protocols are very difficult to
regulated.
implement.
Yes.
It's a very nice talk. I have one question. In this plasma interactions, let's say plasma liquid interaction, we're trying to do the diagnostic. So one aspect what you were mentioning is there is challenges in modeling. We are lacking the cross-section data and other things. Now, how challenging is the measurement itself? Because when spectroscopy is the perhaps the best choice to do diagnostic in these cases. If you do the measurement, you're not doing like a normal air kind, or let's say when your probe is actually coming through some kind of a medium. Here, there is a possibility that if you change your probe from where you are probing the emission, let's say it's coming either through the liquid or through the air, then there will be some kind of additional things or contributions. How challenging measurements are in probing this interaction?
I mean, emission measurements are extremely challenging in my opinion. So, I mean, I've done a lot of work on this in the past, and we often move, if we can, we do laser diagnostics, because exactly why you say. So the problem with emission, of course, is you capture, and it's ultimately line of sight. So forget even about air or other things you're capturing, but it's line of sight. So if you have plasmas with gradients, your plasma need to be extremely stable to do, for example, an Abel conversion if you want to resolve even gradients in the plasma, and this can be already a challenge. Now, I think there are huge amount of opportunities for emission measurements, and the reason I kind of maybe I should have mentioned that when addressing this. Laser diagnostics also have their limitations, particular in species density, limits or thresholds you can measure, even in electrons. So if the EDF, you want to, for example, understand what the tail of the distribution is doing, maybe in some plasmas we are getting to the sensitivity of Thomson scattering to be doing, and maybe in some plasmas, but I don't see that happening for many plasmas in the near future. And I think, for this kind of processes, I think emission might be important to get a better understanding or an easier accessibility to electron kinetics. Particularly, through, for example, excited states that have a very high energy, but even with a few electrons, that you radiatively could capture information about this. I think that particular in, while some of these things has been addressed, and maybe, argon or some noble gases, moving some of that insights into molecular gases, I think might be an opportunity. And, your question, I agree there are a lot of challenges if you look to complicated systems, but I mean, who is telling you to look to a complicated system to start? Okay, I always have some doubts about that. You can look to a very simple plasma or some of the challenges of gradients or some of the challenges of having put it in a chamber, nice chamber, you fill the chamber completely with helium or argon, so you can capture it without these interferences. But I think, there, if you could combine this with modeling in a simple system and trying to understand really the kinetics of some of these transitions, maybe typically for higher energy, we can learn more about the plasma dynamics. And I think that could be quite important.
If I'm allowed, I'll ask one more. Yeah. When we do this plasma mediated processing, let's say even with low temperature plasmas, there are sometimes products which is kind of tricky to handle. Let's say if we are just operating in ambient air and we want to avoid ozone, all those can be used. But since it's in that environment, if you're continuously operating, then you are producing a lot of ozone because you're operating in ambient environment. So is there a way we can utilize some material or some other kind of hybrid approach to reduce such things? Or some?
Well, I mean, okay, so if you don't want to produce ozone, you need to go to a hotter plasma. And so, you can kind of choose to, human hot plasma will produce nitrites and hydrogen peroxide. So then you don't produce it. If you do need the cold plasma, I think the only way is, sucking it away or having some active carbon, that is removing the ozone. I just, well, it's intrinsically, okay, it's a very efficient three-body process that, I mean, ozonizers are plasmas, okay? This is probably one of the biggest success stories in plasmas. It works well, okay? So it's to not do this is very difficult. I think you mediate it or you go to higher temperature. These are the two solutions, only two solutions, I think.
Thank you, Peter.
Okay, I think we have a question from Darshit Singh, who raised a hand. Maybe he wants to ask himself. Darshit.
So may I ask the question?
Yes, yeah, please.
Sir, I want to ask about the laser matter interaction and importantly, laser-induced plasma. So how can we sustain the plasma life in vacuum chamber as we did in tokamak?
Yeah. Can we
I'm hearing, so, okay.
Yeah,
so this is a question about laser-induced plasma. So I'm not an expert in laser-induced plasmas, but so what I can say is the following. I'm always trying to avoid it, okay, with the laser diagnostics we do. But ultimately, laser-produced plasmas will be transient in nature if your laser is transient. I mean, your laser is transient in nature. So the lifetime of these plasmas probably depends on the density. Of course, could be much longer than the laser pulse, but ultimately probably will decay on, I don't know, microsecond timescales, ultimately. So, there are ways to probably enhance that time. So I'm not an expert in how to do this continuously, but you could apply electric fields across the gap that could sustain some longer ionization, no? Because you have already a lot of electrons. So you could combine some electrical driven plasma where maybe the laser produced plasma is this kind of the ignition of the plasma and then try to sustain it. I mean, there are plasmas that are maybe not self-sustained, that are self-sustained because, there is a laser, triggering these plasmas to be ignited. But just with the laser, I don't know. I think if the laser is transient, it will be intrinsically a transient plasma. And the best you probably can do is just extend its lifetime, maybe with electric fields. But there might be other experts that have a better answer. Yeah.
So what about we do in lips in liquid? Then we have sample in liquid, so we can't apply the electric field there, or we can, or in the air, atmospheric.
Well, I mean, so liquid is challenging, okay? And so we actually study, I didn't mention that, but we did study discharges in liquid. And so typically if you produce a discharge in liquid electrically, it will also be pulsed. Now, this being said, okay, we need a huge electric field to generate a discharge in liquid. I mean, also you need significant laser energy to generate that plasma. However, once you have the plasma, you form a vapor bubble. So the threshold for ionization becomes much less. So you could imagine that with a relatively modest electric field, after you generate a bubble with your laser-induced plasma, you could actually sustain a plasma in liquid with an electric field. You know that even these fields are not high enough that would generate a plasma in the electric field in the liquid. Sorry. This is actually a problem when you generate them electrically, because once you have like a spark gap, once you have a bubble there, if you applied a huge electric field that you initially needed to generate a plasma, you have this huge arcing going on, okay, because the voltage is not needed once you have a vapor bubble. And so there might be ways of extending its lifetime this way, that does not require huge electric fields, actually, if you apply this in a smart way and, moderate electric field.
Okay, sir. Thank you.
Okay, there is a question from Shika. Is it possible to treat water in large scale with the plasma? What can be the challenge in large quantities?
Yeah, so thank you for the question. So, we do this, yeah. So on a municipal water plant, ozone generators are plasma generators. They treat large amount of water on a city scale. I think the question is probably if the water is direct contact with the plasma. No, so that's kind of a different situation. I think if this kind of DBD-like, and there are companies that have done some work to do water treatment. Certainly in Japan, there are companies doing that. To some scale, where you can treat significant amount of water, but that's not probably, city scale. That being said, I think that the technology is there. Okay, I don't think it is implemented. I think it's implemented on smaller scale, but the technology is there because we have, for example, DBD generators for ozone generators that are very high power. We have it for polymer treatment. So polymer treatment, corona treatment uses DBDs, and so I think the power levels you need are there. I don't think it has been broadly implemented.
Okay, I think there is a question which is related to your answer right now.
Okay, yeah.
Can we apply plasma treatment in polymers? If yes, then what will happen?
Yeah, so this is actually an industrial process. So polymer treatment, they call it corona treatment. I think there are companies in Germany, I think probably who are leading, the field in making these power supplies. The reason why they do that is, of course, for enhancing adhesion. So you could think about, if you have polymers or polymer films that you want to put a coating on, or you want to print something on, like letters or ink or something, typically people treat that with plasma to enhance the adhesion. And, so that's the reason. So what physically happens in these kind of treatments that often happen in air and an oxidizing environment is that you oxidize the surface. Because of that, polymer becomes often more hydrophilic, and then the adhesion can enhance from certain water-based dyes, for example. The other thing that happens is if you have a lot of radicals, you can have etching effects. So the polymer surface could also become more rough. So there is also a structural effect that could enhance that adhesion process. So these, I think, are the two most common impacts of plasma oxidizing, air-containing plasmas on polymers.
Okay, thank you. So now another question from Ananda. Okay, so this is Dr. Professor. Thank you for insightful presentation. In general, if a plasma application project require effort towards both fundamental understanding and simultaneous evolution of the product for that particular application, any advice on how to plan studies and a timeline towards accomplishing such a such a product?
Yeah, this is the $1 million question, is it? I think is generally very difficult. So I can give you the challenge in the US, which might be different in India. So we have different funding agencies that support different kind of plasma research. So there are funding agencies that are focusing more on the fundamental science. And so they are interested, of course, that this would lead to applications, but their time frame of developing a product is not at the end of the project. So they are really looking into, do we enhance our understanding, and could this potentially lead in the future to new technology? So their outcome is not a working project of, let's say, polymer treatment, what we just discussed. Of course, there are many other funding agencies that are more mission driven. Meaning, for example, Department of Defense or Department of Agriculture, that are really funding you because they are interested in decontaminating food. Okay, and so this kind of projects, in my opinion, have been very, very different. So and typically what I have tried to do, okay, is that I got funding from agencies that are interested in science, and then claiming and doing also, with the motivation of understanding plasma biointeractions and the very detailed mechanisms of what's going on. Ultimately, the Department of Agriculture has less interest in that. They want me to make a device and show that with energy that is maybe not more than UV, I can inactivate these viruses on food substrates. And so the scope is very different. And so the reason that we have been able to bridge, sometimes really the application area and the fundamental area is that I had both funding to do the fundamental science underpinning that application, and even the applied funding for trying to make a device that actually shows it's working. So in the US, I think it would be to date, and we kind of tried actually in the decadal study that was mentioned to try to convince agencies to talk to each other about this, because I think there might be beneficial if you get larger teams, to do something like this. But in US, this is very difficult. Okay, there are kind of different funding streams. So I don't know how this is in India, but I could imagine that there are also different people are interested in different aspects of that. And it's, if somebody is really applied and mission driven, and it's you need to be very, it's challenging to convince them that fundamental science is sometimes helping there, because then they easily would say, well, this is not the kind of area that I'm supporting. Yeah. So it's you need, if you can convince people that I think this is important, and our understandings are helping us to optimize systems or choosing what kind of plasma source we would be using for certain applications, that without this fundamental understanding, we would just try out and spend a lot of time and effort. But it's sometimes very difficult to motivate this to funders that are really strongly application driven. So I think you probably need 2 funding streams for that. And the timeline is typically you start fundamentally, you get some preliminary data, that this might be important in the fundamental project, and then in the meanwhile, hopefully with that data, you can try then as a follow-up or maybe slightly parallel, but that's kind of extending towards a more applied area once you understand the science. But that's the ideal situation, but doesn't always work.
Okay, thank you. Yeah, I think we have taken enough questions. Probably we can close it right now. Some questions are also there, I think. It's already 8:30, I guess. So now maybe, thank you very much, Peter, for the wonderful talk. And it was nice to hear about different applications. I myself had some questions, but I think it is already taking us so much time.
I think it's absolutely fine. If people have questions, they can contact me by email. Yes. I can maybe show my email. I'm happy to answer, further questions. Okay, so you have my email here. So please feel free to contact me if you have further questions.
Thank you very much. Yeah. So this is actually the last talk in the colloquium series, and we are going to close the colloquium series with this talk, and it has been really nice to end with such a wonderful talk. So maybe I think Professor Deshmukh may want to speak something.
Sir, you need to unmute yourself.
Sir, you're muted. Could you please?
So thank you very much, Professor Bruggeman, for a wonderful talk. You have spent great time with us and stimulated a lot of interest amongst many in the audience this evening. So we will certainly like to have you for a personal visit sometime as early as possible to Tirupati, as soon
Thank you
as
very much.
circumstances permit. So in the meantime, very many thanks from all of us, and have a great day. It's your day is just beginning in the US, and I think for us in India, it's about time for dinner. So thank you very much, and goodbye, everybody.
Thank you.
Thank you. Thank you.
Goodbye.
Many thanks to all the Camost coordinators and scientists who really put up this Colloquium series together. So thanks to everybody. Goodbye.
Thank you, sir. Bye. Bye.
Bye.